everyone, welcome to my YouTube channel Bio-Patashala. In this video, I will be solving some questions which came in the year 2022 DBT BET JRF exam. So let's start our session. The first question for the day is the cytosine residues in the DNA are methylated at specific dinucleotide sequences. Which among these methylation motifs are found in predominantly in cases of protein coding genes in the plants? Here it stands for ATC. As we all know that animals methylation will occur at island of guanine, that means CPG. But in the plants it is different. In the plant, DNA methylation occurs in all the contexts of all the CPG and CPHPG and CPHPH dinucleotide sequences, which are located at 5' to 3' linear DNA sequence with the cytosine and guanine nucleotide, which are adjacent to one or base part. And these land plants have much higher levels of DNA methylation at CPG and CHG context. So here the answer is option 1. Next question. And during second question for the day, during signal transduction, one molecule of phosphatidyl PIP2 cleaved into one molecule of IP3 inositol triphosphate and diglycerol by the enzyme phospholipase C. Here the mechanism of signal transduction pathway is there. You can see here. Here the phospholipase C acting as a enzyme to cleave PIP2 into IP3 and DAG. So here the option 4 is correct. The next question, the melting temperature of DNA is higher when the content of GC base pairs is higher, because G and C will make bonds with 3 hydrogen bonds. To break the 3 hydrogen bonds, the melting temperature should be high. So the answer here is option 2, GC base pairs are higher. We'll move to next question. What will be the length of the DNA molecule containing 10 million base pair? This is the simplest question. As we all know, 10 base pairs is equals to 34 angstroms, right? So here if you observe, 1 angstrom is equals to, they have asked actually in the millimeters. So we have to convert first 1 angstrom into millimeters, that is 10 to the power of minus 7 millimeters. And for 34 angstrom, how much it will be? 3.4 into 10 to the power of minus 6 millimeters. So we now we got to know how much it would be in millimeters. 10 base pairs is equal to 34 angstrom, that is equals to 3.4 into 10 to the power of minus 6 millimeters. For 10,000 base pairs, sorry, not 10,000, this is 1000 million base pairs, sorry, 10 million base pairs, it is 0.34 millimeters. So the option here is fourth option. Moving to the next question, eukaryotic RNA polymerase transcribes RNA polymerase 2 transcribes mRNA and siRNA and microRNA. These 3 are transcribed by RNA polymerase 2. Let me know in the comment section that RNA polymerase 1 transcribes which type of genes. Okay. The next question is, in eukaryotes, nucleosomes comprise which one of the following histones? As we all know, histone is an octamer. It comprises of 5 types of histones. The H1 is located outside and 4 complexes are paired. That is H2A, H2B, H3 and H4. So here the option is 4, second option. The motif commonly found in the core promoter of protein coding genes are BRT and Cossack sequence. consensus sequence, because in the core promoter to carry protein genes or on picking box, here it is BRE and the Cossack consensus sequence. So here the option is 3. The introduction of nonpolar molecule into water results in, as they are not soluble in the water, because toluene sometimes it will be in a solid form, and it is when we put it to expose it to the temperature or room temperature, it will form into a liquid. So it won't form any type of solubility in the water. So when it is not forming any type of solubility in the water, obviously there is a decrease in the entropy. If it is forming any type of solubility in the water, then it will form an increase in the entropy of the water. So the option is a decrease in the entropy of the water. And question number 9, last but not the least, for transcribing protein coding is the RNA polymerase forms the pre-initiation complex. So before transcribing a protein from mRNA to cytosol, it need some kind of complex. So what are that complex? Here the mechanism is there, how it will form RNA polymerase to all these things. If you clearly go through it, first D is binding to the sequence, and A is attaching to the D and B, and again F is binding here, and E and H forming the complex over here. So the option here, what are the complexes involved? Option 3. And what are the gap junctions? Question number 10. Gap junctions are absent in. These gap junctions, when we will see, means when there is a gap between two cells. So here brain cells would have gaps, so that's why they would they would make these gap junctions fill that gap. In the cardiac muscle cells also there, in liver cells also there. These are non-motile. These become stationary. But in the erythrocytes, when we observe, these will be moving in the blood, so they don't need any type of gap junctions. And sperm cells also, they don't have gap junctions. This is another example for gap junctions. So for this, today the session is completed. If you need further questions, let me know. I will upload the video regarding DBT BET 2022 question papers and all the necessary topics to all of us. Okay. Thanks for watching. Please kindly share, subscribe, and comment below what are the topics needed for things. Thank you.